Thank you for watching another video on reverse engineering of pharmaceutical dosage form. Today I'm going to talk about lecithin, and because of its complexity I decided to make them in multiple parts. So today we are watching part 1. So what is lecithin? Lecithin is a mixture of different organic molecules. It has phospholipids, it has glycolipids, it has triglycerides, got free fatty acids, it has complex sugar like polysaccharides, monodisaccharides, it has moisture, it has minor organic molecules, and depending on the source and the process, the composition of these molecules will change and vary in the lecithin. In this slide we have two different types of lecithin. One of them is the standard liquid lecithin and the other one is de-oiled lecithin. And we see the percent weight per weight of these different molecules in the mix. Now the difference between de-oiled lecithin is that they will take the standard liquid lecithin and they remove all the components that are soluble in acetone, and then they convert the liquid lecithin to some solid material which they call it de-oiled. And in the process mostly the triglycerides are removed. The liquid lecithin, as the name says, it is liquid or viscous liquid at room temperature, and the de-oiled is a solid, like a sticky, gooey solid, and they are sold in either granulated form or in the powdered form. There are different types of lecithin other than these two available. They call it hydrogenated lecithin or fractionated lecithin. So it is not just limited to these two. These are the molecular structure of some of the compounds available in the lecithin. We have triglycerides on top. We have two types of phospholipids, the PC, which is phosphatidylcholine in the middle, and also PE, which is phosphatidylethanolamine. We had one of the glycolipids at the bottom. They call it DC, and all these molecules, we can look at them, they share the fatty acid which is attached to either glycerin or some sort of phosphor or disaccharide group which is hydrophobic. So we have a lipophilic component which is the fatty acid, and we have the hydrophilic component which is the other, like either phosphor or disaccharide or sugar or glycerin. This is the slide I borrowed from one of the supplier of lecithin, that it shows the application of this excipient in pharmaceutical dosage form. And the picture at the bottom, we have the liquid lecithin at the left and the de-oiled powder lecithin to the right. The application of lecithin is mostly in food industry. It's an emulsifying agent which is food grade and is an excellent source of essential fatty acids. And the lecithin application is mostly in food industry, not in pharmaceutical. And to best of my knowledge, lecithin as an emulsifying agent is not the first choice of any formulator. It is a difficult excipient to work with, and it has a limited application, I would say. So it is not widely used as an emulsifying agent in pharmaceutical. But regardless, there are some dosage forms that they use lecithin as an emulsifying agent. There is a monograph in USP Pharmacopoeia for lecithin, and the definition is what I just said, that it is a mixture of phospholipids, glycolipids, and other components. There are tests in the USP Pharmacopoeia, and one of the interesting one is the HPLC with the ELSC detector that looks at the phospholipid content. And this is rather new method addition to the pharmacopoeia, and in the earlier publication, the USP Pharmacopoeia, this test was not present, it was absent. For the qualitative and quantitative analysis, there are methods with the HPLC UV, HPLC ELSC with all reverse phase, normal phase, and reverse phase. But today I'm going to talk about the GC with USP 401 method. Now, in the USP 401 method, the approach is that we take the lecithin and we do base hydrolysis followed by esterification. So in this process, we break off all the fatty acids from all these different components in the lecithin, and we make an ester out of them. And we analyze these molecules, the fatty acids, with GC, and we do determine the total fatty content. And this way we will determine the amount. Also, we will get information about the grade of lecithin. This is a GC chromatogram of the USP 17A peak ID solution. The USP monograph talks about DB wax column, but to generate this chromatography, I use DB624. We see the myristic acid, like a C14 fatty acid, around 12 minutes. C16 or palmitic acid, it is around, let's say, 18 minutes. We have group of C18s, the saturated stearic acid and unsaturated oleic acid, and the linoleic acid and linoleic acid. These are the monounsaturated, diunsaturated, and triunsaturated fatty acid. There's a group of C18s. Then we have C20, C22s, and on at the high retention time. This is the chromatogram of the one type of de-oiled lecithin, and I believe this is from soya. And I have my internal standard peak around 12 minutes. And the only fatty acid that the soya de-oiled lecithin has is a C16, which is palmitic acid, and the C18s, the saturated stearic, and the mono-di-triunsaturated oleic, linoleic, and linoic acid. So with this chromatography, we can do the amount determination, and also we can get some information about the grade, given that you use right reference material for this work. The next is a liquid lecithin. Again, the source is soya, and is very similar to the previous chromatography. And we only see C16 and a group of C18s. This is a special lecithin, which is fractionated lecithin, and also it has MCT. And I believe this is from soya. We see C16, group of C18, and then the C8 and the C10, which are caprylic and capric acid, that comes from the MCT that present in the lecithin. This is a nice table provided that I borrowed from one of the lecithin supplier that talks about composition of fatty acids in lecithin from different sources. So by looking at your fatty acid that you run your GC, you can get information about if the lecithin in the formulation that you are reverse engineering is coming from soya, or is it from sunflower, or is it the egg. So you can not only get information about the amount of lecithin, with using this chart or this sort of publication, you can get information about the grade of lecithin. This is the last slide almost that I'm showing you FDA IID database. The lecithin is mentioned in number of different dosage form: topical, oral, rectal, vaginal creams. And there are 3 different types of lecithin mentioned. The regular lecithin, the soya bean lecithin, and hydrogenated soya bean lecithin. So these 3 types. The references, I've used NF monograph and the USP 401, and I usually don't mention the suppliers, but I make an exception today. And also I want to make reference to the American lecithin company, that they have good information available in the website. Thank you very much for watching this video. If you like it, please click the like button, and if you like to see more videos. I appreciate you subscribe, and please do write comments, and I'm really looking forward to read your comments and learn from you. And I learn from your experience working with lecithin and any other information that you provide. Thank you very much and have a good day. Bye.